Hello students. Today in this video we are going to discuss about the asexual reproduction in amoeba and yeast, and there is one experiment in your syllabus in which we have to study the asexual reproduction in both through a slide. Okay, so what are the various steps, what are the material required, what is the observation that we are going to discuss in this video. Okay, so in asexual reproduction, we know that binary fission is one of the mode of asexual reproduction in case of amoeba and some other organisms like plasmodium, okay, euglena. So in order to see the binary fission, what are the materials that are required? We need a microscope, slide, okay, prepared slide we need, okay. Now procedure, how we do the experiment. So we'll take the slide and place it on the stage of the compound microscope. Okay, here this portion is called the stage of the microscope. This microscope is compound microscope. Okay, so we'll place the slide over the stage and then we'll observe the slide. So when we see the slide under the microscope, okay, we'll see the amoeba, okay, which is not dividing. The resting phase or before the division of the amoeba, we'll see that amoeba possesses a single nucleus with the cytoplasm, okay. So observation when we take, okay, if we keep the if we take the living amoeba under the microscope, okay, if we take the fresh water or we take the water from a stagnant water body, then we'll see that the nucleus slowly start dividing. First it get elongated, you can see. Slowly the division of the nucleus starts. This is called karyokinesis. Okay, so this is karyokinesis. The division of the nucleus is karyokinesis. Then now you can see the cytoplasm is also dividing slowly. So the division of cytoplasm is called cytokinesis. Okay, see here, after the division of the nucleus, both the nucleus they have moved in the opposite directions, okay, and the cytoplasm has divided, and as soon as the division is completed, two daughter amoeba are formed from a single mother amoeba, okay. So two nucleus, cell membrane and cytoplasm, okay. Now what we conclude from this experiment, we conclude that a single amoeba can give rise to the two daughter amoeba, okay, by the process of binary fission, in which we can see nucleus, then there is cell membrane and the cytoplasm, okay. Now next we will do the budding in yeast, okay. Now material required are same. We need a compound microscope and a prepared slide. procedure. You can see we have placed the slide under the microscope, okay, and then we'll observe the slide under the microscope. Now you can see the yeast over here, okay, this is yeast. Now now in case of yeast, what will happen, the nucleus will divide everywhere. The nucleus, the genetic material has to divide, okay, because it carry the information. So nucleus will divide and there will be formation of some outgrowth, okay, and in that outgrowth the nuclear material will move. So here what you will see is, see, outgrowth has formed, okay, nucleus has started dividing, okay, the yeast is present and here the nucleus has started dividing and outgrowth is there. So this nucleus will migrate to this part, okay. You can see. See, this portion, if you see, the bud has formed, okay, the outgrowth has taken place. Now again this nucleus which got divided from here, again it is dividing, okay, again further one more bud is formed over here, okay. Okay, so here you can see the process of budding in case of the yeast, okay. Yeast is also known as the baker's yeast. So you can see there is formation of the new buds, okay. What we can conclude here, after budding the formation of the bud had formed and they get detached, okay, or detached from the parent body and new individuals have arisen, or daughter yeast have arisen from the existing mother cell, okay. And in this you can see vacuole is present, okay, then cytoplasm is over here, cell membrane is present and nucleus is present, okay. So this has produced the 2 or 3 or number of the young individuals, okay, in case of the yeast. Fine. Now let's do budding in the hydra, okay. Hydra is found in the aquatic habitat, okay. You can see a bird-like structure has come out, okay. So here you can see a bird, okay. In case of hydra, hydra has 2 layers, okay. 2 layers are present, that is ectoderm and the gastroderm. So in some portion the food material get accumulated, okay, and there is division in the cell and outgrowth formation take place, okay. There is outgrowth, and this outgrowth, okay, that you can see, okay, it develops as a bird in case of the hydra, okay. So here you can see 2 layers are present, that is epidermis and gastrodermis, or you can say endodermis, okay. Now bird-like structure has formed, nucleus is migrated, okay. The genetic material that is present, one portion or small portion of the genetic material is also given to the bird, and this after some time this bird it get matured and it get detached from the parent body, okay. Okay, as you can see, it is growing in size. Okay, and as soon as it gets matured, okay, there is detachment. Okay, so tentacle development take place, and in between tentacle there is the oral cavity or mouth is present. Okay, and as soon as it get matured, so here from here it get detached from the parent body. Okay, see, it get detached and get attached with the substratum, any support, okay, that is present. Okay, so this is how the young hydra is formed by the process of budding. Okay, so this experiment you can go through, and okay, if you have any problem you can let me know.